This video is on nucleotides and proteins. This is a pretty important AP biology topic and pretty interesting. So let's take a look. So we start with protein synthesis for prokaryotes. Like all other living things, prokaryotes need protein for structure and function. Proteins make up everything about us. They give cells structure, allow them to move, make energy, and join together to make multicellular organisms like us. We're going to be looking at the process of making protein in prokaryotes, which is called protein synthesis. There are two major steps in protein synthesis, and that's transcription and translation. Transcription is the process of copying DNA, the master instructions for the cell, into another molecule called messenger RNA. Cells don't want to move their DNA around all the time, so when they need the instructions to make protein, they make a copy of their DNA to use temporarily, which is messenger RNA. After they're done making the protein, your cell can get rid of the messenger RNA. If it needs to make the protein again, it can go make another copy. In prokaryotes, transcription occurs in the cytoplasm, which is made of a jelly-like substance. RNA polymerase is the protein that actually does the copying of DNA. DNA is one long strand in prokaryotes, but within that long strand are different sections called genes. Each gene codes for one protein. In prokaryotes, genes are organized into groups called operons. These genes code for proteins that are used for a common function, like metabolizing the sugar lactose, so they're all grouped together. Translation is the final step on the way from DNA to protein. It is the synthesis of proteins directed by a messenger RNA template. The information contained in the nucleotide sequence of the messenger RNA is read as a three-letter words, which are triplets, and they're called codons. Each word stands for one amino acid. During translation, amino acids are linked together to form a polypeptide chain, which will later be folded into a protein. The translation is dependent on many components, of which two we will look at just because they're extra important. First of all, the ribosome, which is the cellular factory responsible for the protein synthesis. It consists of two different subunits, one small and one large, and is built up from ribosomal RNA and proteins. Inside the ribosome, the amino acids are linked together into a chain through multiple biochemical reactions. The second component is the transfer RNA, a specialized RNA molecule that carries an amino acid at one end and has a triplet of nucleotides, an anticodon, at the other end. The anticodon of a transfer RNA molecule can base pair, which means form chemical bonds, with the messenger RNA's three-letter codon. Therefore, the transfer RNA acts as a translator between messenger RNA and protein by bringing the specific amino acid coded for by the messenger RNA codon. Nucleic acids consist of a chain of linked units called nucleotides. Each nucleotide consists of three subunits, a phosphate group and a sugar, ribose in the case of RNA, deoxyribose in DNA, make up the backbone of the nucleic acid strand, and attached to the sugar is one of a set of nucleobases. The nucleobases are important in base pairing of strands to form higher level secondary and tertiary structures such as the double helix. The possible letters are A, C, G, and T, representing the four nucleotide bases of a DNA strand: adenine, cytosine, guanine, and thymine, covalently linked to a backbone. In the typical case, the sequences are printed next to one another without gaps, as in the sequence AAA, GT, CT, GAC, read left to right in the 5' to 3' direction. With regards to transcription, a sequence is on the coding strand if it has the same order as the transcribed RNA. Okay, and then as we take a look at notation, so you can see we have two notations that have 10 nucleotides. So the first one is AATCCGCTAG, with the second one being AAACCCTTAG. So given the two 10 nucleotide sequences, you will be asked to line them up and compare the differences between them. This means calculate the percent similarity by taking the number of different DNA bases divided by the total number of nucleotides. So in our case, there are three differences between the two 10 nucleotide sequences, and so therefore there are seven that are the same. So we divide 7 by 10 to get 70% similarity, and then to find the difference, we take 100 and subtract that from the 70, and that gives us a 30% difference. The genetic code is a set of rules by which information encoded within genetic material, DNA or messenger RNA sequences, is translated into proteins by living cells. Translation is accomplished by the ribosome, which links amino acids in an order specified by messenger RNA, using transfer RNA molecules to carry amino acids and to read the messenger RNA 3 nucleotides at a time. The genetic code defines how sequences of nucleotide triplets, called codons, specify which amino acid will be added next during protein synthesis. With some exceptions, a 3 nucleotide codon in a nucleic acid sequence specifies a single amino acid. The vast majority of genes are encoded with a single scheme. While the genetic code determines a protein's amino acid sequence, other genomic regions determine when and where these proteins are produced according to various gene regulatory codes. Histones are highly alkaline products found in eukaryotic cell nuclei that package and order the DNA into structural units called nucleosomes. They are the chief protein components of chromatin, acting as spools around which DNA winds and playing a role in gene regulation. Without histones, the unwound DNA in chromosomes would be very long. For example, each Homan diploid cell containing 23 pairs of chromosomes has about 1.8 meters of DNA. Wound on the histones, the diploid cell has about 90 micrometers of chromatin. When the diploid cells are duplicated and condensed during mitosis, the result is about 120 micrometers of chromosomes. A nucleosome is a basic unit of DNA packaging in eukaryotes, consisting of a segment of DNA wound in sequence around 8 histone protein cores. This structure is often compared to thread wrapped around a spool. Nucleosomes form the fundamental repeating units of eukaryotic chromatin, which is used to pack the large eukaryotic genomes into the nucleus while still ensuring appropriate access to it. Nucleosomes are folded through a series of successively higher order structures to eventually form a chromosome. This both compacts DNA and creates an added layer of regulatory control, which ensures correct gene expression. Nucleosomes are thought to carry epigenetically inherited information in the form of covalent modifications of their core histones. And then finally, we have telomeres. A telomere is a region of repetitive nucleotide sequences at each end of a chromosome, which protects the end of the chromosome from deterioration or from fusion with neighboring chromosomes. For vertebrates, the sequence of nucleotides in telomeres is TTA-GGG, with a complementary DNA strand being AAT-CCC, and also with a single-stranded TTA-GGG overhang. This sequence of TTA-GGG is repeated 2,500 times in humans. During chromosome replication, the enzymes that duplicate DNA cannot continue their duplication all the way to the end of a chromosome. So in each duplication, the end of the chromosome is shortened. This is because the synthesis of Okazaki fragments requires RNA primers attaching ahead of the lagging strand. The telomeres are disposable buffers at the ends of chromosomes which are truncated during cell division. Their presence protects the genes before them on the chromosome from being truncated instead.